I'm going to open this first session with a keynote speech. Dr. Anton Nekrutenko is a professor of biochemistry and molecular biology at Penn State University. He's associated with their Center for Comparative Genomics and Bioinformatics, and is particularly well known for co-creating and co-leading the Galaxy Biomedical Analysis Software Project. Galaxy, as we all know it, has well more than 50,000 registered users and more than 10,000 active users, and it's among the leading academic genomics and bioinformatics computational environments. We are honored and delighted that he's able to join us today and share more about his research. Anton?
Well, thank you very much. Can you hear me from here? Yeah. Okay, good. So, because I'm too jittery usually, I cannot speak into the microphone, so I'm going to run around. So, I want to thank you deeply for inviting me here. It's been wonderful. I don't go to Louisiana that often, but I know lots of people here, so this was a great opportunity to talk about Galaxy and also to see lots of people. So again, thank you very, very much. And not the least, I tried greets this morning, and they're actually wonderful. I mean, I think they're really, they have some kind of a bad vibe about them. People don't need them because I think I was the only one. But they're great. So it was all good. So if you look at this title, it's obviously very different from what you had in program. Because when people ask you to submit titles for the program, you copy and paste it from somewhere. And then you actually think about it right before the talk, what you're going to talk about. So it's a fairly vague title, so I'm not quite sure what that means. But what I'm going to talk about, some challenges which we try to solve with Galaxy. I don't see bioinformatics as a field. I think that this is just a part of life sciences. It's integrated part of life sciences. There's no other way to deal with it now. And it's just because it's still sort of early stage of all of this, that's why we somehow cut it out and talk of bioinformatics separately from life sciences. But that's one big thing. It really covers. It's not an unwanted child of biology. It's a really big piece of it. And so one of the depressive things that I'm going to talk today about is general crisis of reproducibility in big data biology. It's not because somebody is guilty of this. It's just, again, because it's new. And there are some things that when people start to deal with big data, they just don't know how to deal with it properly. For many reasons, and we're going to talk about some of those reasons, and again, I'm going to try to talk how we try to solve this through Galaxy. But in general, we are in very bad shape with trying to, for example, replicate our analysis. Okay, we'll just use keyboard. So reproducibility is not a fancy word. There are some other fancy words there, for example, such as provenance, reusability, things like that. But all I mean by reproducibility is that you receive a Nature of Science today, you open it, you see a paper, you go through materials and methods section, you can go to your computer, for example, and actually repeat these analyses exactly. This is all I'm asking. And this is a very, very important, because otherwise there's really no much difference between science and, say, political process. It's very important to repeat, so you can, in fact, see that the claims which were made in the paper are, in fact, true. And so, speaking of science, there was this wonderful illustration. It has nothing to do with biology at all. But I found it very interesting because I could show it to my 12-year-old and actually tell him that, in fact, it's actually less than a quarter of the fuel that you burn actually moves the car. So let's see sort of where the waste is happening. So most of it is heat loss, and even from that mechanical power that you have, still you have quite a bit lost to friction. So in life sciences and life sciences analyses, Reproducibility stands at roughly only about 21% as well. So again, I think this is a very good illustration how wasteful these things are. I don't think that everybody appreciates how ineffective internal combustion engines are, but it's interesting to compare it with the state of affairs in big data biology. So we again can reproduce about 1/5 of the results which are published. This is very important. So there is this new project where a group of people is trying to repeat results in 50 selected cancer papers. And so there's this link, which of course none of you can memorize, but these slides are online. And so these are some of the preliminary data which were generated by this effort. And so what it tells you that about 6 out of 41 papers list what version of software they used, and the same situation as well with tools. And if you divide 32 by 127 or 6 by 41, you'll get about the internal combustion engine efficiency here. So we're really in low numbers with all that. And so I really like these cartoons. Can you imagine that if we would ask for confirmation during political debates? So somebody makes a statement and we actually try to reproduce it. So I think in that case, we should be proud of ourselves. I think 21% would be really good. But nevertheless, this is again a very good cartoon because it essentially shows us that with 21% reproducibility, we're not much different from that. So it's something which needs to be fixed eventually. This is a great example this is a good hashtag, methods matter, you can post that. This is an example of just one tool. This is an example of why it's very important to list which tools you use and which parameters you use these tools with. This is its performance of BWA. BWA is one of the well-used mappers. You take next generation sequencing reads, you map them against the reference genome. And you have y-axis here and you have x-axis here. So on the x-axis, you have versions of the software. And on the y-axis, you have allele frequency for just one variant. And you have 4 lines here because these are combinations of 2 parameters. Whether we use defaults or we change them differently. So you can see that, of course, it plateaus somewhere around version 5.4. It's actually 0.05.4. But nevertheless, it would be really important to know that if you are talking about personalized medicine and somebody goes into your office and you survey allele frequency, well, if you make some kind of a conclusion about their health forecast, you're probably better doing this with 5.7 than with 5.2. So I'm not talking about being correct. I'm talking about recording enough information so you can see why you were wrong in the end. This is why reproducibility, again, is important. I think it's right. Actually, rounding is good, so I will lose like 1,000 calories. Thank you so much. And so, let me give you, I don't know why I like to talk about Science magazine today, but a really fascinating example of 2 science papers. So let's go back to about 1985. So this is one of the first PCR papers. It comes from the lab of Norman Arnheim, which is still there in the University of Southern California. Kerry Loomis is one of the middle authors here. And this is about, they worked on beta-globin genes, and that's where they decided to, instead of doing all this subcloning, let's just amplify it with a set of complementary primers. So this is a figure legend. As you know, Science magazine is a kind of peculiar publication, so they usually put materials and methods in all sorts of inappropriate places, for example, such as figure legends. Because isn't it amazing that the figure legend is actually much bigger than the figure itself? But if you, in fact, read this, you can actually go ahead and, provided that in 1985 you had and ability to synthesize oligos, you can actually go through this, and in the end, you would really be able to reproduce this. Really, this is one of those papers where you read it, when you read this paper, you have two strong feelings. The first feeling is that how come I didn't come up with it myself? Because this is a really elegant way of doing this. And the second, I want to repeat this now. And so you cannot do anything about number one, but you can do quite a bit about number two. This is totally reproducible. And the same thing, if you look at many classical papers, for example, Sanger sequencing paper in 1977, you could also replicate that. Again, provided you can get dideoxynucleotides, which was not that easy in 1977. And now let's flip a little bit further. So this is a classical cancer sequencing paper, and it's not wrong. I don't think they have any wrong results in this paper. No, I don't know, but it doesn't seem to be. So you have many papers like this in Nature Science, Nature Genetics, PLOS Biology, all of these journals. And essentially you have controls, you have people with tumors, you sequence them, you're trying to find which mutations make the affected individual different from controls. But so in this case, if you want to reproduce this, if you have that strong feeling to reproduce these results, or not necessarily reproduce, imagine that you are a cancer biologist as well. You're reading this paper, you also have controls, you also have individuals with cancer, and all you want to do, you want to study that particular cancer, not necessarily the cancer described in this paper. And so what you want is that after reading this paper, you say, okay, I go to materials and methods, and I actually want to replicate what they've done because I'm interested in my tumor, in the tumoral study. So in this case, that's sort of where you go. And so there were stringent criteria. And so when you actually dig to online supplement, you find this with two very helpful links. And of course, these are generic links. They don't point to a particular version of dbSNP or to a particular version of 1,000 genomes. Well, what 1,000 genomes? This is index.html. It doesn't have any biological data there. So we go in sort of this cycle where we'll look up the paper, we go to methods, we go to online methods, and so it's a never-ending cycle. So it's not good. And again, I probably seem angry to you. I seem pessimistic to you, but there is really nobody to be angry at here. It's not happening because these are bad people on this slaughter line. It's just there's really no established procedures. It's not easy to make things reproducible. Well, some people appreciate it more than others. And there are some really sad examples. For example, the Annual Louisiana example from Duke, where the results which were published were complete statistical nonsense, in which case, and the problem with this was that there were actually clinical trials based on these results. There were patients recruited, but in the end, this ended into essentially yielded obviously nothing. But the interesting thing is that in some cases, you really have sort of a black changing to white and white changing to black, with some of the test scores changing from 5 to 95 and things like that. So if you would have, or reviewers had access to mechanics of how this study was done, this wouldn't happen. So it's kind of hard to understand how it came through peer review process, but since most of the people here, I assume, reviewed papers once, twice, 3 times, 4 times, 5 times, we all know, well, p-value was smaller than that, so it was good. So next sentence. But the bottom line is that don't trust black boxes in the sense that we need to know how the underlying analysis were done. So this is really easy to complain. It's really easy to criticize papers of others, especially if they're in science, because you're jealous. You also want to have papers in science and Nature and so on. So they publish in science, therefore they're bad people, or they have it easy. But okay, is what I'm saying actually achievable? Is it actually possible to do reproducible science? Well, that's a very big question. And what I'm trying to argue here if we solve this problem, because to many of you in this audience, reproducibility might not sound like a big deal, because for many of you are, I assume, practical people. So you want to map your reads, you want to get the answers, you want to get results from your RNA-seq experiments, you want to know what lives in the gut of somebody by doing metagenomics, things like that. You're not particularly worrying about reproducibility. But what I will argue is that if we solve problems related to that, we would actually solve lots of practical problems as we do this. So this is a very good goal, to solve multiple problems that we have in life sciences, because life sciences have become so data heavy recently. So we'll try to see, so in the next 3 hours, because I have 123 slides. So sorry, the next speaker, I apologize. So we're going to try to answer these questions. So who are we dealing with in this data-driven biomedical, biological? You know, if you write NIH grants, it's going to be biomedical. If you write NSF grants, it's going to be biological. So who are we dealing with in this enterprise? So we have users, and by this I mean people who just want to get their analysis done. They are, say, experimental biologists. I should note, I have a really bad English day, maybe because of grades, I don't know. As you recognize, I'm a goddamn foreigner, so I cannot speak proper English. But this is not unique to biology. There are lots of other disciplines which have the same problem. But let's talk about biology. So these are people who want to do biology. They have critical knowledge how to generate a hypothesis, how to get them tested. They are drivers of the biomedical progress. We have developers, and these are people who like to write code. They write tools. We have, so HPC stands for high performance computing. It's just I'm restricted by circle here, so if I spell it out, it won't look pretty. But HPC stands, this is clusters, infrastructure, compute infrastructure. And so in this circle, we're talking about people who provide HPC. For example, in Louisiana here, I assume there are computational facilities managed by people. There are probably maybe statewide facilities. There are clouds, commercial clouds, the public clouds, things like that. They all go into this category. So there are 3 categories of people, if you will, involved in this process. People who want to do biology, people who want to write code, and the people who administer big compute. And they all want to get funded. So the users want to get their grants because Illumina sequencing is expensive. Developers want to get their funding because they don't want to deal with paperwork and so on. So they just want to live free and write code, and I understand. HPC people want to live because they require heating, cooling, sysadmin, and they need to buy new boxes every 3 years. Plus they have to pay for networking and things like this. So they all have very similar boring interests, such as getting funded. We also have very similar high interest into advancing knowledge. So these will be 3 painful slides in terms of background. So what do users want? They suffer from data logistics. You all know that when you get FASTQ file from somebody, it really depends what it really means. They suffer from HPC trouble in the sense that we are not physicists. So physicists are trained to deal with Unix shell, but we're not. And that's one of the problems that will probably be taken care of as time progresses, as the curricula change at universities. We will become as physicists eventually, so this problem will probably go away. They usually have poor knowledge of existing tools, and that's again not because they're bad, because lots of tools are published in journals like Bioinformatics, for example. It's a good journal published by Oxford University Press, but nobody really reads it. It's one of those, it's clearly not users, because you open an issue, it has 20 application notes, and who knows, and all of them provide links to GitHub. What is GitHub exactly? GitHub, in a good case, sometimes they still source forged use, but anyway. So they know what they want, but they often don't quite know how to develop these tools. And of course, all of that really results in the lack of reproducibility in the end. So they have their problems. And these are the problems when you talk about bioinformatics, you hear a lot about this. But that's not the only important group of people here. Let's talk about developers. So developers, they want to write code. They don't necessarily appreciate the big picture, the big biological picture. So they sometimes use tools for a specific, for example, workflow, specific analysis workflow. But they don't really expose it wide enough, so it doesn't get cited as much. Many tools that you all use, they have parameters. You need to decide how many mismatches per read, or what is, if you're doing RNA-seq, what's the minimal isoform fraction? How do you know these things a priori? There's no idea. So how do developers convey to users which parameters to use? Well, they don't want to deal with users to start with. I mean, good luck writing to a developer. They have other things to deal with. Functional tests don't pass. Again, the data format nightmare in the sense that when formats are not well defined, no new formats which will be generated. And finally, one problem for developers is that it's not as, I'm not saying it's easy, but it's more difficult for them to get high-profile publications and publications of their methodology because journals that are even supposedly computational, such as PLOS Bio, they're not published anything if it does not have a sound biological question. And sound biological questions for developers usually come from their biological collaborators, so it's their thing. There are some, of course, successful examples. For example, the Salzburg's group in Hopkins, they probably have bow tie, tuxedo suite, so they know how to do this. But again, they have lots of collaborators. But usually, it ends up in those journals, good journals, but the journals which biologists don't read. And finally, for HPC, they would be happy to get NIH funding. That's where the money is. Because most of the existing high-throughput computing infrastructure, such as, for example, XC framework, it's a big national compute framework, they're funded by NSF. They would like to have biological users, but biological users don't know how to use them. So there's this misalignment. And a lot of the software written by these people is really not optimized for HPC. It's parallelized on a per node basis, but it's not really. And is it fault of developers? No, because this field moving so fast that there is really no point in tuning something to the extent that it can run on all clusters, only to discover 2 months that, in 2 months that, for example, nanopore sequencing has become a new standard. You need to rewrite all the tools. So it's still a very much evolving thing. And HPC in general is heterogeneous in the sense that you might have university clusters which run one brand of Linux and period, and support only one scheduling system. You can have clouds like Amazon which don't care what you run. So you need to adopt, and it's again, when HPC providers look for biologists, it's hard for biologists to adapt. So that's the 3 groups. And what I'm going to argue, of course, because it's my baby, is that Galaxy solves all these problems. That's the only solution you need. And no, I'm quite critical of our efforts as well, but it's a keynote, so I have to sell. So when I looked through, when I went through all these problems with these 3 communities, you hopefully saw that there's lots of overlap between them. And so if we somehow can tackle this overlap, then we can perhaps achieve something. So this is, if you've never seen Galaxy before, that's its interface. It's the public site. So it has tools, a center pane where we sell things as well, or where the 2 interfaces appear. And importantly, it has a history. And history, that's the part which essentially is responsible for its reproducibility features. You record all steps of your analysis in Galaxy. And so it's a free, we distribute Galaxy under academic free license. So it doesn't matter who you are, university or commercial entity, you can take and do whatever you want with it, never talk to us again. So it's sort of a free crack kind of approach. Again, it's open source software. Academic free license is one of the most permissive licenses. It's very extensible, so Galaxy in itself has nothing to do with biology, really. So there are galaxies for meteorology research, for sociology studies, even for cosmology, which is kind of cool. So Galaxy doesn't know anything about biology until you plug it with relevant data types, tools, and things like that, which would analyze data that you want to analyze. And so some of the ideological goals is how can we make this relevant tools on the relevant hardware accessible to biologists, how to enable communication of the analysis. So you can actually, people who want to build on your methods to advance their own field, to advance their own types of questions can do that. And again, reproducibility is one of the key issues here. But these are, of course, ideological goals. The practical goals is, again, how to get users what they want, how to hook tool developers, how to convey to them that it's a good idea to develop tools for Galaxy, because Galaxy team cannot do this. We cannot wrap all the possible tools. And another goal is Galaxy needs to run on any HPC, at least on all the common flavors of high-performance computing infrastructure. And so from the standpoint, so you can also use the world sort of looks like this, but with Galaxy, we're trying to make it a little bit more predictable. So we're trying to make it sort of a boring Tetris. So everything is really falling into place. But right now it is more or less like this. So how does this work? So again, Galaxy by itself doesn't know anything about biology or what to do with data. So we populate it with tools, and there is a language, an XML, a syntax, which describes tool behavior. And as I'll show, it's very expressive. So this interface, that's the analysis environment, which does not force you to record anything. It just does it for you. So it records everything which is happening by itself. It has a workflow system which allows you to chain analysis and to make a workflow. So instead of a tool, you have now this meta aggregation of tools, which again produces some kind of a predetermined result. And we're trying to develop various ways for sharing and publishing results, so making them accessible. So you can export these modules, import them perhaps in your Galaxy, and doing all sorts of things. So this is very much, so data federation goal, I'll mention it, is a big future goal of ours, of the project. And of course, we also see visualization as the key future way we're going to analyze data, because right now, the typical mode of analysis is like this. I get my gazillion reads from core facility, I ran something on them. I got my result, I ran something on them, waited for 2 weeks, I got something from them, I look at it, and it doesn't look good. It doesn't look good because you didn't set parameters correctly. So you set parameters correctly, you run for another 2 weeks, and you say, not good. So you run analysis correctly, and so it's a kind of an endless exercise. But if you do it through a kind of a visual analytics approach, where you can tweak parameters and see how that affects subsets of your data you know something about, and then rerun this on a whole data set, that really makes it a much more interactive experience to analyze your data. And this is our new graphing framework. You can make some beautiful graphs with Galaxy, and this has been published previously. This is a way to sweep through parameters. So what you see on this picture here, this is cufflinks. So cufflinks, it's one of the RNA-seq tools which assembles transcripts from spaced alignments from band files. And it has parameters. But again, when you run cufflinks, you have no idea what these parameters should be. You really don't know. And so essentially you can create a parameter tree where you're essentially instructing Galaxy to go through all possible combinations of the parameters. And for each combination, it generates in this case, it generates a transcript model. So you can look at that transcript model and say, well, that looks that looks reasonable, so you can run it on the entire data set. These kinds of approaches, and this is Cufflink's example, but it's all pluggable, so it would work for any other tool. And Galaxy runs on a variety of, so we talked about interface a little bit, talked about tools just a bit, but then you need to run it somewhere. And you can either use our public website, which actually runs out of Texas. It runs at the Texas Advanced Computing Center. Just state we're no longer able to sustain no load, and this is TAC is a good facility. You can install it locally, and if you have already existing resource managers, so they're essentially tools which manage your clusters, it will work with many of them. Galaxy right now at TAC works with Slurm. Or you can deploy it on the cloud. It's using the CloudMen. It's a little tool which essentially is designed to orchestrate initially Amazon clusters. You can use it without Galaxy just to create clusters of Amazon machines. It will also work on the Google Compute Engine. The Nectar is an Australia-specific scientific cloud in Australia. And so iPlant Atmosphere Cloud is also one of the clouds we are working with. But I should say one thing about iPlant is that iPlant is the resource which allows us to run our public instance of Galaxy at Texas. Essentially, they pay for our allocation, so it's a very good collaboration between them and us. So what is happening now is that sequencing is becoming progressively cheap. So many labs have their own MySeqs, many facilities have HiSeqs. So who knows what the next sequencing technology will be? Illumina is, of course, is the top earner right now in that field, but that can change very quickly. And the reality of this is that we're going to have to deal with situations where we have many data sets to analyze. It's easier to analyze a few data sets with Galaxy, but when you have 1,000, are you going to click 1,000 times? So it's sort of becoming a little bit unscalable with the current interface. And so what I'm going to do right now is that I'm going to sort of go back. I'm going to try to spread these target communities a little bit. Although in this case, these are no longer communities. These are classes of problems that we have to deal with. User interface, how to encourage developers, and how to leverage HPC. So again, what I mentioned right now is that we're going to have many data sets. And Galaxy user interface does not scale well when you have, again, RNA-seq across many individuals with, say, 3 controls and 3 treatments, or ChIP-seq experiments on a large scale. It's really hard to scale in an existing version of Galaxy interface. And so we're trying to develop new ways to sort of visualize your user space in Galaxy. And this is one of the examples. This is a multi-history view. So the history in Galaxy, it's as you generate data sets, you build them in your history. But you can have multiple histories. And it's kind of difficult to look at them at the same time. So this is one of the new features allowing to do this. This is fully draggable and scrollable type of interface. You can search here. Another feature here is that if you run a tool, so for example, you need to run cufflinks on 20 data sets, or you want to run PWA on 100 data sets, you have this multiple data set button where you can click and select 100 data sets. And what this would instruct Galaxy to do is just take and run PWA on all these data sets with just one click. But one of the really fundamentally different concepts which we're still working on, some of it is already in production, but it's a hard problem, but it's a very interesting problem. It's data set collections. So what data set collections are, I guess it's best explained here. So when you get your data from your facility, again, I'm dealing with sequencing data here because that's the data I'm familiar with. But this can be mass spec data, or it can be, air traffic control over Baton Rouge at different time points. Whatever data you have. So what I can do, I can condense all these data sets into a data set collection. So I will have just one thing in my history. But if I expand it, I will see that it's a collection of paired data sets. So this is paired end sequencing. So this is the sequencing where I have forward reads and reverse reads, or it could be, it's paired end could be made pair sequencing. So it's a paired example where I have a collection of data sets from an experiment, but in that collection, I have relationship between data sets. And so I can represent them like this. So instead of having 8 things to click on, I'm just going to have only one. And within that one thing, all this information that I have isolate, for example, 1141, with forward and reverse reads, is preserved. But the beauty of this is that when you run this, for example, with a tool, the tool will only see this. So you only have to select one thing in the interface, but it can contain 1,000 data sets. But it will go and do this for 1,000 paired data sets. So an example, this is a kind of a structured collection where you have forward and reverse reads. You can have unstructured lists. There are some challenges associated with that. Is that if you know anything about sequence analysis, you know that there's a read groups. There are all sorts of ways of labeling individual data sets. We're just starting to work on how to implement this. But in the end, you're starting to have these workflows where you can see that you have this, how do you call them in electrical engineering, these wire strips, the flat wires and ribbon cables. How? Ribbon cables, yes. So instead of single connections like the needles like this, you're starting to have ribbon cables. So I have a collection here. This collection has, say, 1,000 data sets. And I do one operation on it, and then I will feed all results of that operation to, say, a bow tie, which is a mapper. And then I'm going to merge them all. So I have this kind of a reduction step here, which would condense all these data sets. And in the end, I will have just one. So if there was 1,000 inputs, you can see how complicated this image would look like. But the data set collections is just one thing. And this is an example of full-blown RNA-seq analysis with control and treatment, and that's the reference genome, and so there are lots of ribbon cables here. And this is an analysis on, I don't know how many samples. This is, the good thing it's not from us, it's from somebody in I think in Minnesota or Wisconsin, where they already leveraged the collections and created workflows like this. So I'm showing lots of complicated pictures, talking lots of strange words with a horrible accent, but the bottom line is that we're trying to make interface scale up to a very large number of samples. So this is one of the really important goals because number of samples will grow. Right now we're sequencing genomes, and we think that's a big achievement. But once we pass that stage, we'll be sequencing transcriptomes for every cell type at various stages, ChIP-seq for this, ChIP-seq for that. Conformational analysis, you're going to have sort of a Mike Snyder set of data sets for each of us, and then there will be no choice. We'll have to deal with multiple data sets. So the goal is 10,000 samples. So genome 10,000K. So people increasingly use workflows. Galaxy sometimes is viewed as a workflow engine. It's not. Workflow is just a nice addition on it. And so workflows are increasingly becoming a unit of analysis. So say tuxedo suite workflow where you're doing entire RNA-seq. You feed it an input, it gives you an output. And there are all these tools in the middle that you operate with this entire workflow as with one thing. And the important, so this is an example, for example, cancer-related RNA-seq workflow. It's a very complicated kind of workflow. So the good thing about workflows is that it's potentially gives you a reproducibility advantage. You can share them. You can send your workflow to a user. But one of the fundamental problems with this is that workflows consist of tools. So if you send your workflows to your friend, you're not only supposed to send, the relationship, what link is what and which parameters, but also underlying binaries, otherwise underlying tools, underlying executables, underlying programs. Because otherwise, you're really not gaining much. You're gaining sort of the linkage, but you're not gaining real reproducibility in the sense that you can repeat everything. And so for this, another of visions of the future is that the central thing in this Galaxy ecosystem becomes not the Galaxy instances, but this thing we call toolshed. And the toolshed, you can think of it, although my co-conspirator James Taylor really hates this term, I really like this term. It's kind of an app store. So it's an app store where you put all the tools there. And then and they have unique IDs. And as you share tools or workflows or histories across many galaxies, main Galaxy instance, Galaxy instance at LSU, Galaxy instance on cloud, they all talk to this and they know, okay, to execute this workflow, I need a particular version of cufflinks and I can get it there. So this is, and toolshed is really about making tool development for Galaxy convenient for developers as well, because we as a team just cannot plug all the tools. So before I'm going to tell you a little bit more about toolshed, I just want to show some examples of how easy it is to integrate tools in Galaxy. And if you like looking at XML, although that would be a little strange. But anyway, I colored it so it looks a little bit better. So Galaxy has a very expressive definition syntax in the sense that you can have tools but very complicated interfaces. Lots of, so I guess I can maybe show you an example. So you can have something like, so you can have interface where you can, for example, you can have different types of parameter settings. So if you want default, but you can show everything possible you can have. So there are lots of these, when you wrap tools, there are situations, for example, when some parameters don't make any sense unless some of other parameters was selected. And so Galaxy 2 interface really, Galaxy 2 syntax really geared well for doing things like this. So for example, you have conditionals. Well, this is a bunch of XML, which is blue. And this is what it actually translates into. Imagine that you want to build a graph, and you have a hard choice to make. You want to plot it as lines, or you want to plot it as points. It's a hard choice to make. But depending on how you make this choice, then there is a whole group of other parameters which won't make any sense, for example, for points. For example, if you choose lines, then line can be solid, or it can be black, and there is a line width. But if you choose circles, then the line width wouldn't make any sense. So through using conditionals, you can see you can flip between, if you choose a line, then you can have solid line with a line width of 1. But if you choose points, then of course they can be circles, and there's a point scale now. So you can flip between these two. And all of this is described in this conditional statement. You can also have repeats. So for example, in this case, it's a tool where I'm trying to group, it's a statistical grouping. I'm trying to compute mean in column 1, but then I think, well, I also want to compute standard deviation in column 2. So I click on the insert operation and it adds things as it goes. And so again, it's a fairly short XML where you add all the possibilities, for example, median and standard deviation. So mean and standard deviation. There's also dynamic options. This is a very powerful feature where you perform operation on an input, but you want to know something about this input. For example, here I'm trying to analyze a human variant file, and in this data set, I have data for 3 individuals, individual 1, individual 2, individual 3. So what dynamic options allow you to do, they look at the input and they convey information from that input file to be displayed in the interface. So I can make this choice. And so there are many other things like this. So the toolshed is our app store. This is where we want people to put tools. It's essentially a version control system. It's going to be a GitHub-based version control system or Git-based version control system. So the tools are owned by people who contribute them, and you can install tools into Galaxy instance directly from there. The important thing about this is that it allows you to manage underlying dependencies. So for example, when you want to install Bowtie, yes, you want to get a file which describes to Galaxy how Bowtie behaves, but you also want to get Bowtie itself. So it allows this. And so this has been fairly successful. So this is the number of repositories, unique owners, and the number of times the tools were cloned from there. So this is going okay. It's not going stellarly well for some of the reasons, but which I will describe, which it will go stellarly well once we implement that. So the way this works is that suppose you install Galaxy, it has no tools, not a very useful Galaxy instance, but from Galaxy directly, you can go to this toolshed. It will go to an interface directly. You can select the tool that you want. In this case, for example, BWA. Again, it's a mapper which maps reads to the genome. You can say preview and install. You can click the install to Galaxy link, and it will go ahead, install it into that Galaxy instance. And now you will get this in your tool pane. So you don't need to go and compile anything. It will take care of all of this automatically. But there are some challenges. And we made a survey of developers. So what sort of they hate the most about the toolshed. And the functional tests and documentation. That's the things that they hate the most. So we decided to change completely how we develop toolshed, and there are many things that we've done. We moved it to Git. This immediately, just in the first 3 weeks, we started getting community contributions. So contributions from people who we don't know, and that's the best thing. We also try to, for example, all the tools have now citation sections. So if you write a tool and you would like Galaxy people to acknowledge you in citations, there's a way to do that. There's also a whole set of tools which allow people, which simplifies the process of tool deployment for Galaxy. It's called Planemo. It's a nice project. I don't know how much time I have, like 5 minutes? Huh? Okay. So the problem, one of the challenges is that packaging dependency is fairly complicated, but we are going to do this with existing package managers. So again, one of our future goals is to use increased use of virtualization. This is an example of Galaxy on Amazon Cloud, and there is a layer which we call CloudMan. And in this case, these boxes that you see, these are cluster nodes on a virtual Amazon cluster. So what it allows you to do, the CloudMan tool, it allows you to orchestrate something which looks like a cluster, but consisting of Amazon instances. Or it's not really specific to Amazon, it can work on other cloud providers as well. And so this is how Galaxy on the cloud looks like. And there are some interesting experiments with the privacy certified Galaxy instances which utilize that. They use these locked Galaxy virtual machines in the cloud. Although it's a rabbit hole, the certification process is something very complicated. And the good thing about this is that you can take a snapshot, which is essentially a virtual machine, which would encapsulate all the tools, all the data, everything you've done in it. And that's a kind of example of ultimate reproducibility. This is a thing that you can instantiate and see everything. I'm probably going to skip through the dependency management. Dependency management is, again, it's a question. If somebody wants to install Bowtie or cufflinks on your own Galaxy, how do you get underlying programs? This is how you manage dependency. I'm going to mention Docker. It's recently a very popular virtualization solution, which is lighter than virtual machines. And it allows you essentially to ship tools in a quasi-safe way. Safety is a relative concept, but it's a safe way of sandboxing. So these tools cannot do much damage to the Galaxy file system. And you can essentially encapsulate tools as Docker containers, and that makes it really easy to put them into toolshed and to install them from the toolshed. But one of the things which I want to finish with is one of the things I'm really excited about, and this is about doing ad hoc analysis in Galaxy. What this means is that you do some analysis, and you use all the tools, and in the end you get to have the limited data. But you have an idea. What if I correlate, for example, A with B? But there is no tool like this in Galaxy for that particular type of correlation that you want to do. So you kind of, what do you do at this point? Well, you download data, put it into, well, worst case scenario, Excel, and you sort of start doing the analysis. So this addresses the key limitation is that even if all tool developers in the world work for Galaxy, sort of put tools into Galaxy, we still won't be able to cover all the analysis because scientists are creative after all. And so the grand experiment which we're doing right now is with using Docker, incorporating of IPython into Galaxy. And IPython is a programming environment which is very attractive because it has a kind of a built-in reproducibility. You can save IPython playbook, not the playbooks, but I forgot how it's called. It's called
Notebooks.
Notebooks, yeah. Playbooks are an answer. So imagine you're doing RNA-seq analysis, and you come to a point where that's where it ends. This is as much as you can do with Top Hat, with couplings. But you can click on this link here, and it's a visualization button. This is Galaxy Datasets visualization button. You click on this link, and it brings IPython inside Galaxy. And you can start typing commands here. Of course, this is designed toward people who know how to do scripting. But again, with changing of curricula, I think we'll see more and more of that. So maybe it's not for classically biological user, but it's a very helpful utility for bioinformaticians, for example, for core facilities, for bioinformatics service centers. And so what you're trying, you get data set 72 from Galaxy history, and so you import, for example, in this case, R functions. You're essentially doing all these kinds of ad hoc creative operations, but within Galaxy, taking advantage of existing history. And you can, this is RNA-seq visualizations. So this is, for example, the number of differentially expressed genes, various kinds of visualizations which you can do natively through R functions. And in the end, you can just say, put this particular data set, and what it would do to put it back into Galaxy. So these are these heat map. The resolutions probably you don't see it from the back, but you do your analysis in IPython and then you put it back, and you can put the entire notebook back as well as a data set. So that really takes care of instead of writing tools for every possible case, you can just allow this. So I'm going to skip visualizations. And I'm just going to mention that we have a really nice pluggable visualization framework. Pluggable meaning you can add new visualizations just like you add tools. And our infrastructure right now, as I mentioned, we're mostly running at Tech, but we can utilize a lot of shared resources provided through Exceed, which is a big nationwide high-performance computing network. There's a new Jetstream project which has just got funded, which would provide a Galaxy with even more computer sources. And I just want to finish with saying that I just write emails, so I don't know, I don't do much. And so it's sort of always unfair that you're inviting me. In reality, you need to invite them. It's more expensive though. But still, so we mostly write, so the custodians of the project, we mostly write emails and grants. But all this work is done by the core crew, which is a wonderful crew. It's split between Penn State, Hopkins, some people are still in, so it's a very actually distributed group. And also our external collaborators who just came out of nowhere and decided to help us, and they're again distributed across multiple sites. And if you want to learn more, we have annual conferences, and one of these conferences will be, so they alternate between Europe and the US, and this year it's going to be in Norwich, and we're going to run, we started running hackathons last year, so it's a really vibrant community event. And again, thank you very much for inviting me, and sorry for going over time. I apologize for that.
Time for some questions for Anton?
Yes.
So one of the things I was curious about was in your addressing the data set collections.
Yes.
I was wondering, how do those collections, first of all, are they immutable once you do processing with them?
They are, you mean, well, once you do processing of them, you get a new collection, so they're immutable as all history items in Galaxy.
Right. So I guess that pretty much answers the other question, which is whether or not, the system would do with changes in the collection. It sounds like that they can do one.
Yes, which is a problem from the standpoint of the disk space.
Right.
But there's, again, if reproducibility is your goal, that's the good thing to do. From the standpoint of running Galaxy at your facility, you can choose to do it differently, but on the main site, we do it this way. Yes.
I have just like a general observation. In my opinion, it was wonderful to hear your talk because you come from a life scientist perspective. I'm a computer scientist by training, so I would argue that some of the problems we have in the bioinformatics community, especially in dealing with reproducibility, is because computer scientists are not trained the same way as life scientists are. Life scientists have this clear goal: this has to be reproducible, and if you replicate something 2 years down the line, it has to run the same. As computer scientists, when we write computer science papers and we look at other people's work, majority of the people never quote a version of their system, or you contact them, can I have access to more specific about this algorithm? They usually don't give you code, or they say, this student has left. So I'm going to argue that majority of the problems that are in the bioinformatics world comes from the computer science world, and I guess it's our fault, I should say it that way.
Great.
Do you agree?
I'll blame computer science for that.
Yeah, it's okay.
This gives me a perfect opportunity. I think that to some extent you're right, because as you all know, whoever have iPhone knows that once you install a new version of OS, of iOS, you're like, God, I want the previous version, but you can't get it anymore. So yes, I understand that. And in general, for example, when you deal with package managers, many of them don't keep the old versions. I agree, but I think the stakes are higher in life sciences in a sense that I think reproducibility is probably an achievable goal. But at least through these new, if you keep, VMs of everything, probably to some extent, that's what we're trying to do. But I agree with you that there's blame on two sides, but I'm not a computer scientist. I tend to blame my group of people. But thank you for suggesting that I have to restrict it.
In a certain way, as biologists, we sort of have gotten used to this issue of plate tectonics under our feet. Anybody who has looked at genome sequences like 10, 12 years ago realized that with every update that the NIH was proudly putting out, all of our sequence coordinates were wrong again because somebody found an indel, 2 million base pairs down the line, and everything else on chromosome 17 was wrong. And so, in fact, there was a petition back then to tell the NIH to please not update every 2 months because you cannot work like that. But in the end, I think we just have to get used to those changing data worlds, and important. I think it's going to be the responsibility of biologists to look beyond, the SNP.
Well, thank you I did not raise the issue of data versions, but it's as important because again.
Yeah, but that goes hand in hand with the analysis versions.
Yes, but
I've recently plucked out a ChIP-seq data set, and that was done on mouse version 9. Well, thank God it's still there.
Well, fortunately, at least the genome versions, they're preserved in a way that you can access them. You can go to Santa Cruz and fetch it.
Yeah, and then you do the translation.
Yes, the lifting coordinates, it's easier to reanalyze the genome in many cases. But as the old saying is that the difference between Windows version and the genome versions is that with every new genome version, the quality of genome actually increases. So, what was the question here?
Yes, it's very interesting to see how the Galaxy has been achieving the scalability, because that's what my original concern on Galaxy approach. So I'm very happy to see some, but still think it's more like task level parallelization, not necessarily data level parallelization, like dealing with big data problem. So do we have any kind of ideal plan to embrace Hadoop or Spark-like approach to deal with really big data?
It's a tool-based question, really. There are very few tools which take advantage of this. So again, Galaxy itself is data sort of agnostic, and do you know of any mainstream bioinformatic tools which, for example, really takes advantage of, for example, MapReduce?
There are some.
There are some.
Yeah, there are some, and I think it's still interesting. For example, Spark provide distribute in-memory data management, right? But on top of that, you can run an existing tools together. I don't know exactly how, but it is doable, I think.
Yes. So we're trying to separate ourselves from the tool development. It's not our. It's not what we're supposed to do. So I think I mentioned that one of the problems is many tools are really not written well for HPC. And it's probably because the field is not stabilized. So there's lots of issues for that. But I'm probably not the best person to talk about this. Well, my co-conspirator would be able to give you much because he's actually a computer scientist who works with scalability. I really need to apologize for going over time. I really apologize.
That's great. No, let's thank Anton again, please.